Excited to talk about the work I've been working on for my PhD, and the title of my talk is The Subfornical Organ is the Nucleus for Peripherally Derived Brain CD4 T Cells Regulating Behavior. And so when we often think of T cells in the brain, I think one phenomenon or one disease that comes into mind is multiple sclerosis, which is an autoimmune disease that is primarily driven by T cells recognizing myelin, a component of the white matter of the brain, infiltrating the spinal cord and the brain itself and causing debilitating neurological symptoms. Outside of MS and this autoinflammatory disease, I think recently there have been also studies demonstrating that aging and also neurodegenerative disease, such as Alzheimer's disease or tauopathies, have increased T cell infiltration. And some of the microglial T interactions that happen there drive the disease progression. But to us, when we were looking at these types of published work and the literature, we understand that pathophysiology is a counterpart of homeostatic physiology. And so, if something is happening in disease, there must be a homeostatic counterpart that has gone awry. And so, if in MS or in neurodegenerative diseases, if we see increased T cell infiltration, there must be, a component or a homeostatic regulatory sort of mechanism where T cells actually are able to coexist in the brain, and somehow this goes awry in a lot of these diseases. But we know nothing about this homeostatic physiology, which led us to wanting to pursue this project. But also, there have also been work sort of pioneered by the Hardy Lab and others that have suggested that infection itself can increase T cell population in the brain itself. So both local infection in the brain and systemic peripheral immunization can seed these long-lived tissue-resident memory T cells that protect the organism from recurrent infection. But outside of this sort of infectious context, post-infection type of context, we often think in the normal state that the T cells and immune cells reside in the meninges or the CSF, cerebral spinal fluid at steady state. And many of the work have demonstrated that T cells exist in the meningeal compartment, dura meninges, and the leptomeninges in the steady state, and also work by one of my PhD advisors, David Hafler's group, is one group that has been looking at T cells in the CSF and have surprisingly found that even in normal sort of healthy human beings, you can find actually T cells in the CSF that have unique transcriptional characteristics. So this led us to be more interested in identifying whether we can actually find T cells in the parenchyma itself, in the microglia. Although they are the most prominent and important immune cell in the brain, there could also be T cells that could be there to support them in some extent. We also were interested because there is prior literature suggesting that CD4 T cells and also the T cell related cytokines have a role in mediating steady state mouse behavior. And so in multiple papers that have used multiple mouse models that are deficient in T cells, particularly CD4 T cells, they found defects in anxiety, depression, kind of developmental, sort of social disorder type of models that CD4 T cells have a role in sort of facilitating this effect. And with them, they have decreased some of the emotional behavior. And in addition, certain cytokines like interferon gamma, which have been shown to mediate social behavior, IL-17, which has been shown to mediate both anxiety and memory type of tasks, and IL-4, which have been demonstrated to sort of important in sort of cognitive and memory tasks as well, have been demonstrated to be able to, yes, modulate some aspect of steady state normal behavior. And a lot of these papers have ascribed the site of these cytokines to be from the meninges. But just highlighting that, T cells and the cytokines that they may release have implications in skewing sort of normal behavior in an sort of not sick state. And so we wanted to challenge the sort of pre-existing dogma that, indeed the meningeal and maybe CSF T cells are the only source of these cytokines and maybe other things that can skew homeostasis in the central nervous system. And rather, there may be certain pools of T cells in the steady state normal brain that can additionally contribute to some of the phenomena that we've been observing for decades. So one of the first things that I set out to do in my PhD with the help of the Gutzendler lab was to set up a protocol to be able to look at whether I can actually find T cells extravascularly in the parenchyma of the brain. So here's a represented image of a CD3, CD4 co-stained T cell that is very distal from the blood vessel here in red in CD31. And we found T cells, although they were very rare, but definitely present in the parenchyma, both in white matter and in gray matter. But then we wanted to ask, is there a distribution or in which the T cells can be found? And so where are they located? So we decided to choose 9 different brain areas in which I could consistently find T cells there, and then take a similar area and a tiled image with a 3D sort of Z stack to be able to identify how many T cells can be found within a similar field of view in each of these 9 brain areas. And additionally, since the meninges are thought to be a place where we can find T cells in steady state, we also took whole mount dura meninges and took 2 different areas in the meninges that we can find T cells and did the similar analysis. And here we can I can say that the subfornical organ is one brain area in which we can actually find a lot of T cells densely distributed. And closely second followed by that is the choroid plexus, but we were very surprised that, and specifically this SFO, the subfornical organ, seemed to be a brain area with high density of T cells, even compared to the meninges, which you can find here, and the white matter in the medulla, which you can also find at a higher level than other areas, but not as much as we can find in the SFO or the choroid plexus. And this is a representative image of what I normally see when I look at the SFO in the brain. So the yellow cells are the T cells, with the red being the blood vessels. But it might be kind of difficult to see where they are spatially. And so we took the exact same image and reconstituted into a 3D kind of, 3D format. And so if you look at some of these yellow cells, even though some of them seem to be kind of within the vessels, when you look at them in the 3D space that's reconstructed from the IAF, so a lot of them tend to be floating kind of distally from the blood vessels themselves, suggesting to us that they're within the brain tissue. And so what's interesting about the subcortical organ, it's actually categorized as a sensory circumventricular organ, and circumventricular organs in general are brain areas that have a leakier blood-brain barrier. And what's also interesting about them is that they're major sites in which they sense peripheral metabolic and sort of physiological state. So the SFO and other circumventricular organs have neuronal cell bodies that have receptors that are able to sense various hormones and sort of molecules such as glucose to be able to regulate the steady state physiology of the organism. And so the SFO is most known to regulate thirst and osmoregulation, so the level of salt in the system. But then they also express a lot of receptors that have to do with metabolic regulation, like insulin and leptin, amylin, that can regulate food intake and energy expenditure. So it was interesting to us that, in particular, like it seems like the T cells that we find are in these SFO, which are able to also sense and integrate a lot of the physiological signals that are important in regulating physiology. And since we did this in mouse model, we also wanted to see if this is also translatable in human. And so with the help of Dr. Marcelo Di Stasio, who pioneered spearheading the Yale Legacy Tissue Donation Program, we were able to get fresh brain tissue from recently deceased donors. And he, because he's a neuropathologist, was also able to meticulously microdissect the SFO and then run IHC on the sections, and specifically for CD3, so that we can broadly look at T cells and to have one, stain so that it's easier to run some of these analysis. But was able to basically regionally find the SFO, the fornix, thalamus, choroid plexus, the neighboring brain areas, look for the CD3 positive T cells in each of the areas, and do a density calculation similar to what I did for the mouse confocal. And we predominantly decided to run these samples on patients who were systemically diagnosed with diseases that are not inflammatory. So it does not have to do with infection or systemic inflammation, where we can expect to find maybe more immune cells infiltrating tissue, but rather many of these patients passed away from like cardiovascular diseases. And regardless of, they passed away, so they're not fully healthy, but we still found that the T cells in the SFO the SFO had the highest density of CD3 positive T cells per brain area, which was reassuring to us that we could find similar observations between the human and the mouse. And so to sort of conclude this part, we found that, we could find CD4 T cells in the brain in, a lot of brain areas, but particularly, surprisingly, found them most densely populated in this one tiny circumventricular organ called the subfornical organ. And then as immunologists, we also wanted to understand, are the T cells we can find in the brain just functionally and transcriptionally unique at baseline, since they're in such a unique tissue like the brain, which is known to be an immune-privileged site. To show this most simply, I wanted to sort of set up, show you the single-cell RNA sequencing experiment, and also go through the pipeline in which I do many of the subsequent flow cytometry analysis. And so in order for us to fully characterize the T cells that are in the brain tissue, we always inject the mouse with the CD45 antibody retroorbitally so that CD45 marks all immune cells, and then we inject it in the blood so that we can label all of the ones that are circulating. And this happens 5 minutes for 5 minutes prior to us taking the mouse and perfusing it and harvesting the tissue. And for these single cell experiments, we took brain, the dura meninges, colon, the white fat, the blood, and then either the mesenteric lymph node or the lung because we had to run it 2 separate times for the sake of science. We also FACS enriched for the T cells by negatively sorting out the myeloid cells or the B cells. And across both of our runs, and the only difference is one has the mesenteric lymph node and one has the lung in blue. When we basically took all of the T cells and then put them on this UMAP space to find out how similar or different they are to each other, we were surprised to find that the brain, the fat, and the meningeal T cells all clustered closely together in the UMAP space compared to the other tissues that are represented, suggesting to us that they are transcriptionally similar. And so we wanted to deep dive into understand, how are they similar and how are they different. But in addition, one way to sort of look at this is to look at T cell state. And so there are many genes that we shown to be ascribed to certain state of the T cell. So we took a representative 3 or 4, representing a naive stem-like, exhausted tissue residence, Th1, Th17, Th2 Treg, and looked for what kind of genes are most highly expressed in the brain T cells themselves. And we found that surprisingly a higher both average expression and the percent of the cells that are expressing it within the tissue was so the ones in the brain most highly expressed tissue residence related genes and also Th1 related genes. So they were highly able to make secrete or express interferon gamma and express genes like CD69 and CXCR6. But then there are certain other tissues that have some of the similar characteristics. Since all of the single cell RNA sequencing is RNA data, we also wanted to validate with protein, and so we stained a lot of these proteins by flow cytometry, and indeed recapitulated the RNA sequencing data and found that a lot of the Th1 associated proteins and tissue resonance associated proteins are highly expressed in the steady state brain CD4 T cells compared to the others. And there are others that I'm not showing here. But one interesting characteristic that we wanted to sort of dive more into was the capacity to be able to secrete the cytokine interferon gamma. So we went about to sort of probe out this 2 ways. So there is a reporter mouse, so a mouse that basically has a YFP protein tied to the interferon gamma RNA. So and so here we found that basically there are a lot of YFP interferon gamma expressing T cells in the brain compared to the meninges, but the white fat is another sort of tissue that has a lot of interferon gamma secreting T cells. And this was similarly recapitulated in the ex vivo stimulation with PMA ionomycin. And this kind of also captures the UMAP experiment where we found that the brain meninges and the white fat all clustered closely together. And we think in part the similarity is due to their high capacity to be able to make the cytokine. And so if we were to take interferon gamma knockout mouse, we wondered if the cytokine is important in the establishment of these T cells themselves. And we're surprised to find that, yeah, in these interferon gamma knockout mice, the steady state number of the CD4 T cells is a lot lower, even though in the periphery it's not really different. And so the cytokine itself is important in the trafficking or some establishment of these T cells in the normal brain. And the transcription factor TBET, which dictates sort of naive CD4 differentiation into Th1s, was also important in the T cell in the brain and normal at homeostasis. And so if you don't have TBET, you also have a lower number of CD4 T cells in the brain. And these, yeah, an entirely lack interferon gamma secreting population as well. So to conclude, we found that the CD4 T cells in the brain are robustly able to secrete interferon gamma, which is and also express tissue resident associated proteins. But some of these traits are more closely similar to what we can find in the meninges and in the white fat, which I'll come back to in a little bit as well. And one thing we were surprised by, because these T cells are able to make the cytokine and also express these tissue resident markers, and from other flow characteristics study and characterizing work, we know that they're memory type T cells. We wanted to that means that they must have undergone some kind of activation somewhere in the body at some point in development. And so we wanted to understand when this sort of T cell establishment and differentiation happens and which tissue might be the source of these brain T cells, or where might be the source in which we can find these Th1 skewed T cells. And so first, to tackle the when question, we looked at mice starting from 2 weeks to look at, at what point in aging or development we can find the brain T cell numbers and cytokine producing fraction kind of level out. And we found that it was at the 4-week period where we found that both the numbers and in the cytokine producing fraction kind of leveled out with the adult mice. And what's interesting about 4 weeks is this is right around the time of weaning. And weaning is when, you take a mouse that's with its mother and separate it so that it can sort of start its own track into adulthood. But developmentally, it's interesting because this is also the time when the microbiota rapidly expands. And so we wanted to understand if this T cell establishment is due to the microbiota expansion that happens during weaning, which can drive a lot of these immune changes, or if it is actually a developmentally timed program, and regardless of the microbiota, happens at this 3 to 4 week stage. And to do that, we got to germ-free mice, so mice that do not have any microbiota, and then looked at this weaning period, weaning reaction in these microbiota deficient mice. And so you can see here that the SPF, sort of the normal facility mice, have this massive expansion with the weaning time point. But then the germ-free mice do not experience the same degree of expansion both in T cell number and in the interferon gamma secreting population. And this suggested to us that it's the microbiota component of the weaning reaction that was driving this T cell establishment. But then we wanted to emphasize that this is not something unique to the brain. In other tissues, such as the white fat, it seems to be we can observe a similar finding. So this weaning-induced expansion of the T cells in the interferon gamma secreting fraction is not necessarily brain-specific, and it happens all across the body. So the microbiota seems to, generally be important for this phenomenon. But one thing we were wondering is the microbiota hosts a lot of different bacteria, and some bacteria we know can skew the immune response. And so, for example, segmented filamentous bacteria, or SFB, is a bacteria that is known to skew CD4 T cells into Th17 subtype. So we wanted to know, if we change the commensal microbiota composition, can we also change the skewing or the flavor of the T cells in the brain? And so when we colonized germ-free mice with SFB, we found a mild increase in the CD4 T cell numbers, but a dramatic increase in the proportion of the cytokine secreting fraction in the brain. And this suggests to us that, not only is it important to have microbiota, but certain species of microbes probably have differential effects in the type of T cells that can eventually seed the brain as well. And I don't have time to kind of go into some of the TCR transgenic ones, but I wanted to go into the question of where the T cells are being trafficking or coming from. And so one way to look at that is T cell receptor sequencing. So T cells are unique in that they express the T cell receptor, and the T cell receptor itself can act as kind of like a barcode, so that we can track where the T cells, if we can find T cell clones that are shared between many different tissues. And so in this single cell experiment that I showed earlier, we also tacked on paired TCR sequencing. And so we also, in addition to sequencing their transcriptome and their RNA, we also sequence the T cell receptor. And so that we can cross-compare the T cell receptor clones in all of the tissue. And one way to represent how likely we can find T cells overlapping between tissues is this Jacquard overlap index. So mathematically, a higher overlap index value suggests higher probability or likelihood of finding T cells that clonally overlap with each other. And I guess we were not surprised that the brain and the meninges had the higher likelihood of clonal overlap because they probably have T cells that are seeing similar brain or CNS antigen. But then I guess we were also surprised that it seemed like there was a lot of clonal sharing between T cells with other tissue, including the fat, which I think since they seem to be transcriptionally similar with the and have high capacity to secrete interferon gamma, I guess maybe there is, and I will show, brain fat crosstalk. But yeah, lung and the colon also seem to have some type of overlapping T cells. And one sort of thing I wanted to highlight too is that we can also actually observe a similar gut-brain T cell trafficking axis in a human and a live human. And so in David Hafler's lab, they were collecting a lot of human samples with people who have donated CSF, colon, duodenum, or blood at various time points. And they had run single-cell RNA-seq with paired TCR sequencing. And in this one healthy human, who's still alive, we actually were surprised to find there are two T cell clones that overlap between the gut and the CSF, suggesting to us that this gut brain overlapping TCR phenomenon is probably not just something that we can see in mice, but we can observe in healthy human. And since this, we have also done a few, and we have seen actually a few more individuals where we can find consistent sort of T cell clones that are overlapping between gut and brain. So it's interesting that we have constant circulating T cells that can seed both tissues. But to more directly address with if we can find direct trafficking, we employed a photoconvertible mouse model in which this mouse expresses this KIK-GR protein expressing KIK-GR protein at baseline, which emits a GFP or green fluorescence protein. But upon shining a UV light to photoactivate it, it can basically convert its GFP to also shine RFP. So we can surgically expose the white fat or the gut and then shine a UV light to basically locally photoconvert this green into red. And so we can track the red cells to see if the fat converted cells can go to other places or the gut converted cells can go to other tissues. And so starting from the gut data, which has also been demonstrated by other groups such as Diane Mathis and Vijay Kutru and EAE model, we found that gut-labeled, photo-labeled T cells can actually traffic to the brain, but also everywhere. It seems like the gut is a source of T cells activation, migration broadly across many tissues, including the brain. But we were surprised to find that when we fat labeled the mouse, we found actually a high proportion of fat labeled cells within the brain compared to spleen, colon, or mesenteric lymph node. So it seemed to be a little bit more unique in that the fat labeled cells being able to migrate to the brain. And in some of the data that I can't show today, it seems like mobilizing the fat by food deprivation is another way to basically be able to push some T cells from the fat to the brain, demonstrating a novel sort of fat-brain axis. But just to highlight that it seems like the T cell receptor data where we found clonal overlap can be recapitulated in this model. To make sure that we can also pharmacologically prevent T cell trafficking into the brain, we know that integrins such as VLA4, LFA1 are important in T cell extravasation and infiltration into tissue. And so during this weaning reaction time point where we can see the T cells established in the brain, we treated the mouse with these integrin blockade antibodies for a week, and we were able to block the T cell establishment in the brain. Suggesting to us that they require, integrins to be able to establish through the circulation. And single blockade did not work. I needed to do double blockade in order for us to see this effect. In addition, we kept seeing CXCR6. It is associated with tissue residency, but we also saw in a CXCR6 global knockout mouse that they had less T cells in the brain to begin with. And so we wondered if this chemokine receptor was also important in the seed establishment, the trafficking aspect of the T cell in the brain. So we employed this adoptive co-transfer method where we took a wild type and the CXCR6 knockout T cells, made an equal mix, and then transferred it into a mouse that has no T cells. And so had the wild type and the CXCR6 knockout T cells compete to occupy the niche in each of these tissues. And so we started out with a relative 50-50 mix, and at the end of 5 weeks, which is how long it takes for us to start to see the T cells in the brain, you can see the majority of the ones that make it to the brain are actually the CD4 5.1 wild type ones. And so basically the ones that lack CXCR6 are unable to establish in this brain or I guess niche, suggesting to us that this chemokine receptor is important in this aspect of trafficking or establishment. To go back to some of the information, the experiments that I had with the SFO, we also wanted to see if the 606 might be important in the positioning within the brain. And so the chemokine ligand for 606 is CXCL16. And so we took basically hole punched different brain areas, the same ones that we looked at in the confocal experiments in the very beginning, and found that the ligand for CXCR6, which is CXCL16, is actually most highly abundant in the SFO containing punch, suggesting to us that maybe there is some aspect of this chemokine ligand receptor interaction that is important in aspects of the density, T cell density, highly associated with the SFO. And so with that, we found that the gastrointestinal and the white fat are places where we can find T cell trafficking from these tissues to the brain, and integrins and CXCR6 seem to be important in the positioning for them to get into the brain and establish. And so the last part, which is one of the most difficult parts, and I think the part that has the most underlying questions, is why are they there? What is the function of this interferon gamma secreting T cells in this normal brain? In order to do this, we had the mice go through a battery of behavioral tests. A lot of them have to do with anxiety or depression because of the emotional behavior and from some of the collaborators that we had in their expertise. And out of doing everything, the only test that had some consistent differences in the T cell manipulated mice was the novelty suppressed feeding test. But unlike the other anxiety tests, this one is unique because it has a food deprivation component to it. So instead of just placing the mouse in an open field, which is sort of scary or aversive for a mouse, there's an additional food deprivation component to it where the mice were food deprived 24 hours before the test. And so the mice are hungry. And then on the day of the test, we place a small piece of food in the center. And so the mice have to balance the survival instinct to, I guess, find and eat the food because it's hungry, while exploring this scary open field environment, because mice are generally timid animals, and so they like to kind of stay at the corners and don't like to be somewhere they're more exposed, like in the middle where the food is. And so consistently in all of the different knockout mice that we had, which lack brain T cells, so the T cell beta knockout mice, the interferon gamma knockout mice, the CXCR6 knockout mice, the T-bet knockout mice, and the smarter rag knockouts, which I didn't have time to kind of talk over today, they take a lot longer to basically find and reach and eat the food in the center, suggesting to us that they are baseline more anxious and have a maybe more difficult time making the decision to go and eat the food in the center when they lack T cells, interferon gamma, or CXCR6. But these are all global knockout mice, and so we wanted to see if it's specifically the cytokine interferon gamma knockout in the CD4s. So I guess I had to skip the slide, but to go over it, essentially, if we adoptively transferred wild type CD4 T cells or interferon gamma knockout CD4 T cells into T cell deficient mice and let them expand, we can rescue the effect. And so if they have the wild type T cells, the TCR beta knockout, the time it takes for them to reach the open field center essentially looks similar to this in that they take a lot less time to be able to reach the open field center. So this led us to be able to conclude that having this interferon gamma in the CD4 T cells is what drives this novelty suppressed feeding test behavior. So to conclude, we think that, and to also go over some of the data that I wasn't able to present today, we think that the interferon gamma receptor on astrocytes might be important in mediating some of this effect, because when we knock out the interferon gamma receptor on an astrocyte-driven Cree, we were able to recapitulate this novelty suppressed feeding defect in behavior. And so, although we don't have the full molecular mechanism and there are a lot of remaining questions here, we think that in some part the interferon gamma cytokine signals to astrocytes, and some of this may have a role in mediating this novelty suppressed feeding test behavior. And lastly, just to put this whole paper into context, I wanted to kind of talk about sort of how I see this work maybe fit in for future work. So there are many ways in which the brain and body can talk to each other. Some of them, the main ones that we know of, include cytokines, hormones, and metabolites. But then it would be interesting if T cells, immune cells, which are very mobile, is another way for the body to be able to communicate and understand what is going on in its physiology. And one interesting sort of reason why this might be possible is that they can offer different amounts of sort of different speeds in the communication in which happens. And so the easiest way for the brain and the body to talk to each other is through direct innervation, which is a very fast response. Cytokines and hormones kind of are medium response time, and they're upregulated fast, but then it takes a longer time for them to circulate and get to the brain, and it involves the adrenal glands usually. And also it can have a more prolonged response. But immune cells could be another way that kind of helps the system adjust to an even more longer time scale of things, where if the microbiota changes, there might be certain things that the body needs to be adjusted to. And so maybe they want to send certain T cells that have various characteristics to secrete cytokines to various tissues, including the brain, to be able to make sure that the system physiology is well adapted to its internal state. But again, this is work that this is a purely, hypothesis-driven sort of schematic, but I think would be interesting to explore further in the future. And with that, I'd like to thank both labs that I've been a part of. A majority of the work that I showed today was done in Andrew Wang's lab at Yale, through all the mouse models and the behavior, all the collaborators who had generously donated their time and their mice to help out with various experiments, and the Hafler lab, which I did all the human work and a lot of the computational analysis I learned from many members of the Hafler lab. And thank you all for your attention, and happy to answer any questions.
Thank you so much, Tomomi. This was a really Tour de France on T cells on the brain. So if anyone has any question, please type it in the